Hello friends, today we are going to study about peptides and proteins. What are peptides? Peptides are the chains of amino acids. Here, two amino acid, the first amino acid and the second amino acids are joined together to form a dipeptide. For the formation of a peptide bond, this bond CONH is a peptide bond. The formation of a peptide bond involves removal of water molecule, that is a dehydration process. A water molecule is removed, one OH from one amino acid and H from another amino acid is removed as H2O and a peptide bond is formed between the two amino acids. Peptide bond is a type of a covalent bond. Now when peptide bond is formed, this terminal where NH3 plus is present, this is N terminal, and this terminal where COO minus is present, this is the C terminal. Because the C terminal of first amino acid and the N terminal of second amino acids are involved in bond formation. So after the formation of a dipeptide molecule, so this is center yellow one is the carboxyl terminal of one amino acid and the amino terminal of another amino acid is already linked together through a peptide bond. So now the free end is N terminal of the first amino acid and C terminal of the second amino acid. So here two amino acids are linked together. That's why this is a dipeptide molecule. What are oligopeptides? When few amino acids are linked together. What are polypeptides? When many amino acids are linked together. Now, after linkage or after the formation of a dipeptide, oligopeptide or polypeptide, the amino acid are known as amino acid residues. They no longer remain amino acid. They became amino acid residue. Why? Because a water molecule is removed. To be in an amino acid state, it should have a N terminal and a C terminal. But after the formation of a bond, the amino acid will lose H2O, due to which it loses OH and H. So it no longer remains a complete amino acid and it became amino acid residues. So amino acid, after they are joined together, we call it as an amino acid residue because of the removal of water molecule. Peptides can be distinguished by their ionization behavior. Now this is an example where four amino acids are linked together. What are those amino acids? Alanine, glutamate, glycine and lysine. Now, if we see, this is the N terminal, this is the C terminal. So these groups can ionize as it ionizes in free amino acids. Apart from those groups, what are this? These groups are now not available for ionization because they already got involved in a peptide bond formation. They are not free now. So it cannot ionize. What about the COO minus and NH3 plus present in the side chain of glutamate and lysine? They can ionize. This is R group. Glutamate is a negatively charged amino acid. Lysine is a positively charged amino acid. So lysine is having one extra NH3 plus group. Glutamate is having one extra COO minus group. So these groups are again ionizable. They can ionize. So they can contribute to the total acid-base behavior of a peptide. Correct? So we can distinguish peptide by their ionization behavior. Every peptides will have different combinations and numbers of amino acids. Depending upon the number and the type of amino acids, their ionization behavior will be different. So we can easily distinguish the peptides based on their ionization behavior. So now for an overall acid and base behavior, what all things we need to remember or we need to check? The free alpha amino group, free alpha carboxyl group, and the nature and the number of ionizable R group, the green one. Depending upon this, every peptides will have different ionization behavior or their pKa value. Now, because of this, the peptides also have a characteristic titration curve as amino acids. Therefore, they have their characteristic pi at which they do not move in an electric field because pi means it is that pH where the net charge is 0. Now, biologically active peptides and polypeptides, they can occur in a vast range of size. There are no fixed size that a peptide or a polypeptide has to be of that size to be biologically active. They can be small in size consisting of only 3 amino acids or they can be larger in size consisting of 25,000 amino acids. These are the examples of peptides consisting of smaller number of amino acid residues. For example, oxytocin. Correct? It is released from the pituitary gland and helps in uterine contraction, consisting of only 9 amino acid residues. Bradykinin. These are anti-inflammatory compounds consisting of only 9 amino acids. Thyrotropin releasing factor, which helps in the release of thyrotropin, consists of only 3 amino acid residues. So these are peptides having less amino acid residues. Still, they are biologically active molecules. Coming to oligopeptides. Insulin helps to lower the blood sugar. It consists of two chains, A and B or alpha and beta. The chain A consists of 30 amino acids and chain B consists of 21 amino acids. Glucagon, which helps to increase the blood glucose level, consists of 29 amino acid residues. Corticotropin, which helps to stimulate adrenal cortex, consists of only 39 amino acid residues. So these are biologically active oligopeptides. Coming to polypeptides, cytochrome C consists of 104 amino acid residues. Chymotrypsinogen consists of 245 amino acid residues. The largest polypeptide is titin. It is a muscle protein, consists of 34,350 residues. So, to be a biologically active molecule, the sizes can vary from 3 amino acids to hundreds and thousands amino acids. Now, some proteins are multi-subunit proteins. Multi-subunit proteins means these are those proteins that have two or more polypeptides associated together. The protein is called oligomeric if it contains at least two identical polypeptides. And those identical units will be called as protomers. For example, hemoglobin. Hemoglobin consists of 4 polypeptide chain, 2 alpha chain and 2 beta chain. So this is an oligomeric protein because it has at least 2 identical polypeptides. So multi-subunit proteins means the proteins that consist of more than one polypeptide chain. Now, when a protein consists of more than one polypeptide chain, those polypeptide chains can be linked together non-covalently as in case of hemoglobin or covalently as in case of insulin. Insulin consists of chain A and chain B. And these two chains are linked together by disulfide bond. So, when a protein consists of more than one polypeptide chains, they can be linked covalently or non-covalently. How we can calculate the number of amino acid residues present in a protein? For that, the formula is molecular weight of a protein divided by 110. Why 110? Because if we take the average of molecular weight of 20 common amino acids, that average will come 138. But all the amino acids are not equally abundant in any protein. So the average molecular weight of most abundant amino acids, if we consider, it will come 128. For the formation of a protein, these amino acids are linked together through peptide bond. And for the peptide bond, water molecule is released. So what is the molecular weight of water? 18. So we have to subtract 18. But all the amino acids are not equally abundant in any protein. So the most abundant amino acids, if we take the average of those amino acids molecular weight, it will come 128. So 128 minus 18, 110. So the molecular weight of any protein, if we divide by 110, we can get the number of amino acid residues that proteins are made up of. Now, the polypeptides have characteristic amino acid composition. All the polypeptides or the polypeptides present in a biological system, they are composed of amino acids, the 20 amino acids, but all the 20 amino acids are not equally abundant in any polypeptide. Some are more abundant, some are rare. So, amino acids that are more abundant or most abundant in any proteins are leucine, alanine, glycine, valine, glutamate, and serine. And the rarest amino acids are tryptophan, cysteine, methionine, and histidine. One important point to notice, the characteristics of an individual protein that depend more on its amino acid sequence than on its amino acid composition. That means the characteristic or the function of a protein or the structure of a protein that depends more on its amino acid sequence rather than its amino acid composition. There are some proteins which contain other groups also apart from amino acid. So the proteins that contain only amino acid residues, those proteins are called simple proteins. If they're associated with other chemical components, we call it as a conjugated proteins. Now, conjugated proteins can be fall into three categories. Lipoproteins, that means proteins associated with lipid. Glycoproteins, proteins associated with carbohydrate. Metalloproteins, proteins associated with metal ions. This table shows classes of proteins, the prosthetic groups attached with those proteins, and their examples. So now one question from my end for you all. You have to tell me why characteristics of an individual protein depend more on its amino acid sequence than on its amino acid composition. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.